Hello everyone and welcome to our first interview session, Science in the Spotlight. We think that science actually has to be communicated, and this is why we as iGEM Munich want to show you some groundbreaking scientific discoveries that inspire us. And this is also why I would like to get a look behind the scenes from our guest professor Jellusova on how research is actually conducted. So let us start with a short introduction. Thank you very much. We're thrilled to have you here, Professor Jellusova. You're a researcher at Technical University of Munich, and you're an expert in immunological research. So especially an expert in focusing on one of the most fascinating and variable cells of our immune system, the B cells. So what makes B cells that special? They're able to recognize several pathogens, foreign substances, toxins that the body has actually never encountered before and protects us against such a variety of diseases. So, since you're the expert, I of course want to ask you, Professor Jellusova, would you like to tell us a bit about your research regarding B cells, and maybe start off with explaining a bit what B cells actually are, and which main function they fulfill in the body?
Yeah, so thank you very much for the introduction, and thank you very much for the invitation to this interview. And so, as you mentioned, B cells are an important part of the immune system, and they protect us from pathogens, which can be viruses, bacteria, worms, toxins. And so the amazing part about B cells is that at any time in our body, we have B cells that can recognize any pathogen there is. And so they protect us by producing antibodies, but they can also fulfill other functions. They can communicate with other cells, and they can affect the activity of other immune cells as well. And so what I'm interested in and what we are studying in my group is basically how B cells process information, how they receive information, and how they make self-aid decisions based on that information.
Amazing. Thank you very much. That sounds very interesting. So you also said, B cells have a lot of functions. The most prevalent that a lot of people probably know is the production of antibodies, so the specific targeting of pathogens or foreign substances. But in your research, you're also specifically focusing on the difference basically between well-functioning, so healthy B cells, and degenerated B cells. So my question would be, how can these degenerated B cells maybe be involved in the development of diseases?
Yeah, so, in every cell makes proteins, and these proteins are encoded by DNA, but if this DNA is altered, the function of these proteins can be also altered. And so that can lead to different behavior of the cells. So there can be cells that are just not able to respond properly to pathogens, and then we're less protected. But then there's other diseases where B cells can contribute to as well. So for example, if B cells make wrong decisions, and if they attack our own body, that can lead to autoimmunity. Mm-hmm. If they attack substances such as pollen or dust, that can lead to the development of allergies. And so basically our research focuses on trying to understand how these different proteins, these signaling molecules, how they change how B cells make decisions.
Okay, fascinating. And now that you mentioned it, you're focusing on maybe the cause of a few diseases like autoimmune diseases, allergies, and basically the cellular mechanisms behind it. So it would also be interesting, just do you think that the mechanisms that you actually discovered as a group, as a researcher, do you think these mechanisms can be important for a medical application in the end, so maybe a treatment for specific diseases?
Yeah, so our research, I would say, is primarily basic research. So we are focusing on the molecular mechanisms, on the very basic processes in the cells. Mm-hmm. But I think ultimately, hopefully, this will lead to either to the identification of new targets that can be targeted in these diseases, or it could help us to understand how these diseases develop in the first place, and perhaps prevent the development of these diseases.
Mm-hmm. I see. Can you maybe name an example or two that come to your mind?
Yeah, so for example, we're working on the molecule that's called GSK3, mm-hmm. and so this is a signaling molecule that is always active in a cell, and when it's not active, the cell actually becomes active, mm-hmm. and so it helps the cell to decide to go from a resting state to dividing and mm-hmm. being more active. And so, the research that we have done in my group has shown that there's a specific subset of B cells that which can give rise to leukemias, which are very sensitive to an inhibition of this molecule. So it was surprising, because you would think if you take an inhibitor and treat the cells, they will actually be more active, they will proliferate more, that that is not what you want in a leukemic setting, right? But instead the cells died. And so you could imagine that this very specific type of leukemia could be treated with an inhibitor of GSK3.
Amazing. So this basic research of underlying mechanisms can have a very big impact, as we can see in your research. So I would be very curious maybe also how you discovered these mechanisms. So maybe also for our audience that is a bit more elaborated in the lab, or has a bit more information about how it actually looks in the lab. Maybe you can give us a few examples of which experiments you actually conducted that gave you these insights on the molecular mechanisms. So which were maybe the major or key experiments? Can you define a few?
Yeah, so we in my lab we usually start with genetically modified B cells. So in this specific scenario, it was B cells that just had the gene for GSK3 deleted, and so they were not producing these proteins. And then we take these B cells and we study different parameters. So we, on a molecular level, we look at the signaling pathways, and we look at how these cells make decisions. We looked at the metabolic level, so what nutrients do they take up, how do they treat these nutrients, and then on the functional level, so how do they survive, how do they divide, how do they respond to when they're stimulated, and then ultimately what it means for overall cell fate decisions.
Okay, very interesting. I think that's already were quite a few insights in your lab. In general, I would be also interested in the fact, because you just dedicated your life, basically your career, to science. So there must be something that you think is particularly fascinating about immunology and B cells. So what is this? What drives you?
Yeah, so I think B cells are so interesting, first of all, because they have so many functions and they affect so many processes in the body. And then also the fact that they can recognize, the population of our B cells can recognize basically any type of pathogen there is, which I think is an amazing accomplishment. And the decisions that the cells have to make are very difficult, very complex, and they have to incorporate information they get from other cells and from their environment. And so all of that, I think, is very fascinating. And honestly, I see them as little, as little personalities which go around, talk to other cells, make decisions.
Yeah, true. It's amazingly complex, and I think it's just there are so many unknown things or mysteries that can still be discovered. And I share that passion for immunology in particular. So it's very nice.
Oh, I'm happy to hear that.
Well, that's why you're here today, right? Yes. Maybe let's just continue here, because we already talked maybe a bit about your career as a researcher. So maybe tell us a bit about how you decided to become a researcher. So what were important steps in that case?
Yeah, so actually I decided to become a scientist when I was 5 years old.
Oh my God, that's very early.
Yeah. Well, everyone kept asking me what I want to do when I grow up, and I thought that's a good answer because I like collecting plants, and I had a little herbarium.
Amazing.
And then when I was 6, my father gave me a microscope, and then I thought that's the best invention humanity has ever made.
Oh my God.
And I never changed my mind. So that's why I decided to become a scientist.
That's very impressive, because I remember, I think I mostly wanted to be a dancer or professional, equestrian or something like this. So I guess it's very unique that you actually knew that at such early age. Amazing. Okay. But maybe relate a bit more about what happened after you grew a bit older, maybe what happened after you were 6, and when you finished your A-levels, when you graduated, what were the main important steps that followed you basically to the point where you are now? So how did you get to where you are today?
Yeah, so I'm originally from Slovakia, but I decided to study in Germany because, Slovakia is a small post-communistic country, so, the scientific landscape isn't as exciting as it is in Germany. And so I did my diploma in Germany, my PhD as well, and then I went for 5 years for a postdoc to the United States, to California, and then came back and started my independent research group and applied for professor positions, and so now I'm here.
That sounds quite, easy if you tell us it this way, because I guess, especially for us as students, it's kind of hard to imagine, or that maybe the path is a bit harder to follow than you might describe it right now. But maybe there are also a few people in our audience who might think about or picture him or herself as a future scientist. So it would be interesting to know, what do you think, what are good qualities of aspiring scientists?
Yeah, so first of all, curiosity. So I think you should be passionate about science, and you should be interested just in how things work, and then I think the most important quality actually is to be stress resistant and to be able to, enjoy science even in a stressful environment, because research is inherently quite stressful. A lot of your experiments will fail, sometimes whole projects fail, sometimes you just don't understand, your results. And then on top of that, academia is very, very competitive. And so if you are able just to put that aside and say, I'm going to do the best I can, but I will enjoy my work and I won't let this stress me out, I think that's the most important ability to have.
Thank you. I guess these are already quite good tips in this regard. So let's see what the future holds, and how our journey actually looks like in that regard. So today we would also like to talk about youth support for our iGEM team. For those of you who might not be familiar with what iGEM actually is, it is an international research competition for students, and it actually takes place in the field of synthetic biology. So we basically try to focus on global challenges, so for example in agriculture or in the medical field, and then try to solve this with genetical engineering. And our iGEM team this year, so iGEM Team Munich, actually focuses on engineering B cells. So that's also why we're here today. It's a type of cell that you now should be very familiar with, since Professor Jellusova elaborated a lot on what B cells actually are. And we want to engineer these B cells by introducing sequences of known antibodies into the cells against specific diseases or pathogens. So the idea is to actually put this B cell back into the human body and make the body basically resistant to those pathogens in the first place. So we can basically engineer our immune system and protect us against a variety of diseases, pathogens, and we're focusing on different targets here. We would be very interested in maybe your opinion also on the iGEM competition. And that is why I want to ask you, which benefits do you think can students gain from a competition like iGEM?
Yeah, so I think these competitions are really great because they give you a more real-life view into how science is being done, and I think it boosts creativity and it boosts independent thinking and troubleshooting and resourcefulness, and then it helps you to work in a team. And then also I think the competitive aspect sort of is, similar to a real-life situation. So I think it's a really great setup.
So at least for learning, it's really helpful. But the question is, do you think that the iGEM project can also have a real scientific impact at some point?
Yeah, I think it can have a real scientific impact. I mean, with the limitation that, of course, projects in immunology or oncology, they usually take a lot of time and a lot of resources. So, the time within the competition is limited, and so that a little bit limits what you can accomplish within this time. But I still think it can, benefit real life research, either because you can refine certain methods, or you can create preliminary data that can be followed up on, or what is really important for scientific research is new ideas, that you can bring new and fresh ideas into the field, and that can inspire people.
That's true, yeah, you're definitely right. I mean, we have basically a period of maybe 5 to maximum 6 months to actually establish a proof of concept in the lab. And if we're taking into consideration that most research projects take at least several years, that's of course a major difference. Although there are a lot of iGEM projects, as you also mentioned, that start off with a good idea and then maybe even go to actually transform that into a startup idea.
Yeah.
But still, I definitely agree with you. And I would also be interested what you actually think about the B-cell engineering in general, since you're the expert in the field. So do you think that this can have an impact, or which impact do you think could the B-cell engineering have in the future?
Yeah, so I think it's definitely a promising strategy, and I think engineering B cells would be beneficial for different types of diseases. So as you said, infections, if you can engineer B cells that would target a very specific pathogen, that would be very helpful, but it could also benefit, B cell engineering could also be useful for cancer, for the development of cancer therapies, or correcting immunodeficiencies. So I think there's a lot of potential. But also, having said that, since B cells do have all these complex roles, it's also quite a difficult task.
Of course. Yeah, I think there's probably a lot of research that still has to be conducted until we can actually go from bench to bedside, basically, with this approach. Although it's just established in mice now, so I guess clinical studies will show how efficient that actually is. Okay, but thank you very much for your opinions on that. So maybe what would be also important if we think about B-cell engineering, because it's maybe not only the feasibility, but also ethical regards that come with it. So what do you think, what would we need to consider if we want to implement B-cell engineering in the clinic, actually, regarding ethical standards?
Yeah, so I think the ethical questions are similar to the questions people in the T-cell field have to face, and so there's a variety of ethical questions that need to be considered. So, for example, you always have to balance the benefits and the drawbacks, and the benefits and drawbacks can be different for different patient groups. Then you have to, when you're developing the therapy, you of course have to, at some point, you have to test your therapy in living organisms, so that would be animal models, and then later on humans in clinical studies. And so, the ethical question is, you of course first have to exhaust the other possibilities and test your method in other models. And the ethical question would be, when do you go into a living organism? And then you of course have to consider which patients you are going to recruit for the clinical studies, but then also what is going to be your patient group for the treatment. And then finally, when the treatment is developed, how do you make it accessible? How do you make sure that also, economically disadvantaged people have access to this treatment?
Yeah, that is also a very good point, of course, because as we know, I think the health insurance actually pays for that in Germany to some extent, but in other countries, as we know, this is still something that people have to be able to afford on their own.
Yeah, and these therapies are really expensive. So,
but also, of course, very individualized and offer very great chances in the future. If we think about just engineering the whole immune system, which is, of course, something maybe a bit of like future music, but yes. I would like to thank you for this very great interview. I think we got a great impression on what your research actually is about, how that impacts also maybe the medical field, so how you go from basic research to ideas that are important for the actual medical application in the end, and also gave us a bit of an insight about your career pathways and the iGEM competition in general. So thank you very much.
Well, thank you. Good luck with your project.
Thanks.